Oi pessoal, tudo bem? Para dar seguimento à nossa série de entrevistas com os pesquisadores e com as pesquisadoras, a gente está trazendo com vocês para o nono IMPB a entrevista com a pesquisadora Ioly. Vamos começar. Hello Ioly, how are you?
I'm very well, thank you for having me here.
I will do some questions. It's a simple talk about genetics, about transcriptomics. It's a interview, but I will try to make a little talk. Okay. First, I have first question. What motivated you to study transcriptomics of fungi by viral? Sorry. When do you realize this was a promising research field?
Okay, I have always been fascinated by viruses because they are between living and non-living entities, and I find that very interesting from a philosophical point of view, if you wish. And I started studying mycoviruses, that's viruses that infect fungi, when I was an undergraduate. And that was because it had never occurred to me that a virus could infect a fungus. And when I learned about it, I thought, oh, what a great idea, that's so interesting, I need to study that.
That's amazing.
And I have always had an appreciation of global understanding of concepts, and that's where transcriptomics comes in. So as an undergraduate, back then I was fascinated by microarrays. I never got to do proper full cell microarrays though, but eventually, once I finished my PhD, which was on hepatitis C virus, I started getting involved in next generation sequencing and what we call transcriptomics in the more modern sense.
Cool. I think this kind of the genetics, it's so amazing because the people use plant, use animal in genetics, but when I think when we study virus, it's a new word to think, to research some things, and I think it have a big ideas to explore this. For the second question, what have been the main challenge you have faced in using transcriptomics in your studies?
Well, the challenges range from sample preparation to RNA quality and bioinformatics interpretation of data. In terms of sample preparation, I think the most challenging experiment we had to do was when we infected plants, plant leaves, with a fungus that contained the virus and the fungus that didn't. So the fungus destroys the surface and the tissue of the leaves, and we were trying to understand how much of the leaf should we use for our transcriptomic studies. We were worried if we were to focus on the damaged area, it will all be dead. But if we were to take the whole leaf and we took all the healthy area, then we may not see the differences we wanted to see. So we had to do quite a few tests to understand how much we should take, how much of the leaf we should take. Because transcriptomics are based on RNA, RNA quality can be a problem.
A problem.
Yes. You know, it's very sensitive,
Yes.
RNA. So we need to be very careful while handling it through extraction and then cDNA synthesis and library construction. We need to work in a very clean environment with fresh equipment, sterilized, maybe filter tips if we can, just to avoid degradation of RNA because of the RNases that all around the environment.
Let's the third question. Which partnership, mentors, or collaborations have most influenced your career and helped you transcriptomic studies achieve great impact?
I am grateful to my mentor, Professor Martin Bach, who actually sent me to a training course to get a better idea how to analyze transcriptomics, because when I started I had no experience, so I was self-taught essentially, and it was good to go to a course to confirm what I had learned myself and also learn new things that I hadn't encountered before. And then I'm very grateful to all the people who gave me their data to analyze, and that includes transcriptomic studies, but also other types of next generation sequencing studies like genomics with my colleague Dr. Esther Garcia-Sala, and microbiome analysis as part of a company.
Ioly, what advice would you give to young researchers interested in transcriptomics, fungus, virus, host pathogen interactions, particularly regarding technical training, model selection, and publishing? What do you give to young researchers interested in this area?
Okay, if we're talking about transcriptomics, I think you should start by doing some RNA work before start doing the transcriptomics work, so you can get a feel of RNA and what you need to do to preserve its integrity, because it's much easier to lose the RNA as compared to genomic DNA or plasmids. And you need to have your lab or your space in the lab ready for RNA work. And that means potentially dedicated pipettes, dedicated material, good cleaning practice, either with ethanol or with commercial solutions that prevent RNase work. You can use RNase-free water or DEPC-treated water that stops the RNases. And you need to do a lot of tests throughout the process to ensure that what you have at the end and you are going to send for analysis maybe to a company is of good quality, because that's where it all starts. If your RNA is degraded, you are not going to get good results. So that's for the wet lab. Then for the computational analysis, I would say you need to understand what each step is doing. I'm not saying you need to know the code, but you need to understand biologically why we do this quality control and what the results of the quality control mean, and why do we do this alignment, and why we align to the genome or the transcriptome, and which software we're going to use and for what reasons. So all this you need to have clear in your mind. There are ready-made pipelines, and there is nothing wrong with using that. But what I advise my students is that they always need to know what they're doing and why they're doing it, and not going blind into it. And about publications, I would say it's very common to see publications where it's obvious the work was done, let's say, by a company. So again, it's important to know what has been done, even if you didn't do it yourself. And be able to write a proper protocol, both about the lab work and the wet lab work and the dry lab work, so that people can follow exactly what you did. So there are many times that it's obvious the authors don't know exactly what happened. They probably gave it all to a company and they analyzed everything, and they say next generation sequencing analysis was done by company X. But you still need to provide information about what the company did.
I think so. But I have other questions. Considering the challenges of RNA-seq technologies, such as biological variability, environmental effects, genome gene and poor annotation in fungal, how does your team address these issues?
There are indeed some challenges in what you mentioned. In particular, in my study of mycoviruses, we know that the environment plays a really great role on the effect the mycovirus will have on the host, because the mycoviruses often modulate pathways, metabolic pathways. So if the fungus has a different carbon source or nitrogen source, the outcome of the infection is going to be different. I would say you need to have a good design and maybe even prepare all your samples together so you minimize the variability of different batches and you can see the effect of the virus in under these specific conditions. And we tend to do that. We tend to try to keep the environment as controlled as possible. Now, fungal genomes and annotation is another big problem. What I normally do once I get the reads and after the quality control, I align them both to the genome and the transcriptome of the fungus. So what we see is that alignment to the genome might be very, very high, like over 90%, 95%. So that's a good alignment. There are always some sequences that are not going to align for various reasons. But the alignment to the transcriptome might be 50%. So there are a lot of transcripts that are not annotated on the fungal genomes. Now, I would like to say that we spend the time to annotate the fungal genome properly, but this hasn't happened yet. We just reported this discrepancy and continued with what was available for the fungus. I know it's not great, but so far we hadn't had the resources and the people to address this more thoroughly.
Yes. In projects where you evaluated transcriptomes and small RNA, such as your work on, I will speak slow, I have problems to say this word, Dostostroma septosporum. Yes.
Very good. Yes, correct.
Which aspects of RNA processing do you consider most promising for uncovering mechanisms of virulence or fungal control?
So we are really keen to do transcriptomics in Dostostroma septosporum, which is a fungus that infects pine trees. So it causes damage on pine trees, and it's quite widespread in the northern hemisphere. By combining transcriptomics and small RNA sequencing, what we want to see is how the fungal defenses against the virus may affect the fungal phenotype as well. So we have our immune system when we get infected by a virus, right? We have the complement, and we have the antibodies, and we have all the immune cells. The fungus doesn't have that. The fungus has RNA silencing. Because these viruses normally have RNA genomes, they can be targeted by the RNA silencing machinery. And there is an enzyme called dicer, which chops the RNA of the virus in small pieces. And then these pieces are used by the RISC complex of Argonaut proteins to guide another RNAs to the viral sequence, and so that the virus can be digested completely. Now, these sequences are very, very small. They're like 18 to 22 maybe nucleotides. That's the sequences generated by dicer. And therefore, they may have spurious similarities to mRNAs of the fungus. So we're looking for off-target silencing, whether the fungus can actually target its own transcripts in the process of targeting the virus, and whether this is going to alter the phenotype of the fungus.
Okay.
Does that make sense?
Yes, yes.
Good.
Thank you. You finished the interview. Thank you so much. It's a big pleasure to stay here, to talk with you. Thank you so much, Ali.
Thank you. I enjoyed it very much. You did a great job.
Thank you.